Hello, everyone, and welcome to our last journal talk this semester. And please, without further delay, Olga, the stage is yours. Welcome.
Okay, hello, everyone. Today I'm going to answer on your questions you posted on YouTube channel. There were not a lot of questions, and so I try to be quick. So the first question, it was, do the authors explain, or you could speculate, what is the relevance of change in the main source of energy? So yes, according to the data they present, we can see that there is a change in respiratory exchange ratio. But authors had really a short explanation for this. They said that this increase in RER was caused by increased food intake, and this chow, mouse 8, they had high amount of carbohydrates. I found this chow they put in the article, and it looks like this. So the explanation is quite easy, and I'm not sure if we can really compare it, but I found the article. Some of authors are the same, and here they were testing. Okay, because for them it was surprising that after MGS treatment and ablating AgRP neurons, mouse seems to stay on the same level with body weight and also with food intake. So they did a little bit longer checking of this mice. In the article they said it was 4 months, and they found something similar, because even with the smaller carbohydrates level, it seems that with increased food and we have increased respiratory exchange ratio, but the energy expenditure is the same. So yeah, this is like the confirmation that it was all about the food intake increase. I'm not sure if it's fine, this answer for you.
So maybe I have missed that. So they don't really have an explanation why there is a switch from one kind of predominant source to another, right? So, like,
yes. They said it was just because of increased food intake.
Oh, okay. So they mean that, okay, since the majority of the food is kind of constitutes by carbohydrates, that's how you get more. Okay, it kind of makes sense. Yeah, I think. Yeah. Okay. Thank you.
Okay. So the next question. The stronger increase of food intake in arcuate lesioned AgRP sold mice suggests a counteraction of arcuate AgRP neurons balancing the hyperphagia and controlling the stiffness. Have the authors obtained some data to support or rule out this mechanism? And unfortunately, they also don't present a mechanism, what they suppose happened. I had, for me, it was a proof for them that this AgRP cells from hindbrain can stimulate feeding independently. But I did some checkout. So first, if we would like to think that AgRP neurons from the hypothalamus are responsible for this, there is a publication about mapping the inputs in the brain, and as you can see here, there is no direct connection between arcuate and the NTS region in the hindbrain. Here they use AAV virus for tracing. Yes?
So, Olga, I meant not that those AgRP neurons in the arcuate kind of project to those in the NTS and somehow hinder them from this. I just found interesting that if we have intact both arcuate and those hindbrains, then there is the effect is kind of weaker than if we would have the ablated AgRP neurons in the arcuate and we have only hindbrains involved. This question is like total speculation for me, but it could be it could be that that maybe the existence of those arcuate AgRP neurons somehow makes the response of AgRP circuits kind of smooth.
Yes. Okay, I think I understand the question this way. Yeah. So the first, I would what I mean to say it was that AgRP neuron from the arcuate can't be like there is no direct connection as between these two group of neurons. So we need to look for something else. And now I have few options for it because we know that there is a connection between POMC neuron in the arcuate and POMC neuron in the NTS. So maybe here somehow they can regulate it. If the AgRP neurons in the hypothalamus are missing, maybe this is the way they can communicate. We also know that arcuate POMC neurons and POMC neurons in the NTS, they have inputs from different parts of brain. So maybe the same as POMC neurons in the NTS, these AgRP neurons in the hindbrain, they also receive some inputs from other part of brain, and this can be the way they can regulate. I mean, still the AgRP neurons in the arcuate are missing, so but maybe in the other part of the brain there is something what can regulate it. And we also know that there is a 12 nuclei that they can that they have connection with POMC neurons in the arcuate. AgRP neurons in the arcuate and also POMC neurons in the NTS, and maybe one of this region can control once again this hindbrain AgRP neurons when AgRP neurons in the arcuate are missing. Also, there are some proofs for this that mechanism of action of POMC neurons in the NTS and POMC neurons in the arcuate looks a little bit different. I mean, POMC neurons in the arcuate, they look that they need a longer time for action, and the POMC neurons in the NTS can act really quick. So maybe also, this is the way how they control these AgRP neurons in the hindbrain in case of AgRP neurons are missing. Yeah, I don't know if you can agree with it.
I don't know. I was asking. So the authors didn't speculate on that. So it's only, as far as I see, that's only your research. But they didn't even discuss that, right? Did they?
Yes. Okay, there is one more thing. I mean, it's also my speculation about it, because there was research that when we did MGS treatment, we can see increased thalassocyte proliferation in this region. And as the researchers claim in our article, this AgRP population in the hindbrain is heterogenic, and they have also classified these cells as thalassocyte-like. So maybe somehow we can have increased thalassocyte proliferation in the hindbrain, and it also makes this cell answer faster for I don't know, somehow. Okay, can we go to the third question?
Please.
Okay, so the third question was about, do you or authors have any explanation, speculation about the lack of food intake increase after stimulation of hindbrain AgRP neurons during the daytime? So for this, author had an explanation that maybe some critical components of the hindbrain AgRP cells is under circadian regulation. We know that in the arcuate AgRP neurons, we have a circadian rhythm. Mifsud told us about it a few weeks ago. So for me, it's, I mean, I agree with this explanation. Maybe if I can say something more, I'm still a little bit confused about this experiment because they claim that they did this measurement in the dark time and in the daytime with the light. But when I start to read the explanation in materials and method, it looks like the stimulation of the AgRP hindbrain neurons, both it was during the daytime. Because they said that here we have a time when they check the food intake, but before this food intake, they stimulate the neurons 90 minutes earlier. Also, mice had some time to spend in the cages, and also like another 90 minutes earlier, they also checked the background food. So I mean, it looks like all this stimulation was did in the daytime, and yeah, it's a little bit confusing for me.
What you mean is that they have this 12-hour cycle, and then their night cycle starts around 7 p.m. So if they do it 90 minutes before, it would be still in daytime. Although
yes.
The measurement, what they do is in the nighttime, but actually the stimulation was done during the daytime, right? So this is...
Yes.
It's really worth checking the details, right? Sometimes when you can discover something very interesting.
Okay, and the last question, why do you think these AgRP neurons were overlooked in previous research? What might be the reason? So I'm not sure if I can make a clean explanation for it, but I did a little checking in, let's say, the history. So in 1988, they already discovered the Pham population in the NTS. I mean, because it was surprising because for a long time we know about the Pham population in the hindbrain, but no one were looking for AgRP neurons. And they found this NTS population by doing some immunocytochemical techniques, and they just found this fiber connection between these two regions. And as you can see, the discovery of AgRP in the hindbrain was almost 10 years later. Also, as you can, as we know, 90% of AgRP neurons co-expressed NPY, and I think that if I read the older publication, they were mostly using NPY to detect AgRP neurons in the hindbrain. So maybe that's why they couldn't find the population in the hindbrain. Also, I think that we just need to wait for more advanced techniques to detect this group of neurons, because they are really small group. And also they are heterogeneous, so maybe even if someone found it before, they wasn't sure what they really had. And I think also it could be the reason that no one cares about this part of the brain, because they were so focused on examination this hypothalamic region.
Do you have the
Yes, and
please go ahead.
In general, so we have chosen this paper to show that usually it is not good to stick with the dogma, and the dogma in this case was AgRP neurons in the arcuate. But my question was also related to the functional, let's say, overlooking. So do you think that if they use the like genetic tools with the AgRP promoter, do they target as well these neurons? So in fact, the effect is a whatever effect is a combination of the both populations of neurons.
Great.
I mean, it must be the effect of both group of neurons, but maybe this if AgRP neurons population is active, this hindbrain population effect is so small that they didn't found it before. And you, what you said about this AgRP promoter?
Because in general, if they use, for example, optogenetics, so they stimulate exclusively these neurons in the arcuate. But if they use the AgRP promoter and, for example, chemogenetics, then they stimulate both populations, right? So this might be the reason that they, and the same is with the ablation. If they use the AgRP promoter to express whatever diphtheria toxin, then in this case they kill the both populations of neurons, right? Giving the, so maybe that's the reason that simply using some tools they killed both, killed or interfere with both populations of neurons, and did not see the effect coming from the hindbrain population. On one hand. On the other hand, it is strange that if they use the specific tool for the arcuate neurons, that they did not see the effect from the hindbrain neurons. But this is what you say, that maybe this is a small population having the really minority function from the minor function from compared to the arcuate neurons. Yeah, that's just the speculation. I don't know, but that's the case, right?
I frankly don't think it seems to be a minor function. Like what they show is that with the ablation of those AgRP neurons within the arcuate, you can still have actually even stronger effect. So even though as Olga says that there is probably less neurons than in the arcuate, that's actually my other side question. Do you have the estimate of how many neurons? Is it about like 2,000-3,000 in the arcuate, and how many in the hindbrain do they alters? Because like using the single cell sequencing, you can actually quantify like how many neurons. So that's an interesting question.
I have
agree with Vijay, sorry, just to finish, that those studies which use those kind of chemogenetic approaches or like the diphtheria toxin-related ablation approaches, we need to kind of return to them at some point and then understand that in these studies, they have actually impacted both populations, both in the AgRP as well as in the hindbrain. And we need just to keep that in mind. They didn't know that at that time, but we need to make this kind of correction in the view of this new data, which is very interesting.
Yeah, Olga, do you know what is the estimate of the number of neurons in the hindbrain?
No. I don't know the numbers, but after reading this article, I cut the brain of our AgRP-expressing mice to look for the GFP signal, and the signal from hindbrain was really weak. I mean, I need to do some GFP staining to be sure, but it looks like it was just few cells in this region visible. So
compared to AgRP, right?
Yes. Mm-hmm.
But at least that's good that you see them in your own experiment. So
yes. I mean, it's quite kind of tricky to cut the brain like this because I need to be extremely careful during taking this brain. But yeah, maybe it's worth it. We will see.
yeah, and one technical explanation is that usually you cut out this part of the brain if you are focused on the arcuate, right? So it's easy to lose them.
Any other questions or comments? Anyone? Pham, Mifsud, Shania, or anybody else? Olga, maybe a few words to conclude. Your critical assessment, anything you found particularly nice or particularly not nice or whatever, like whatever you should say, maybe some conclusion about this paper.
Yeah, okay. I mean, I like this paper. I like this paper because, I mean, it looks like we still can find something new in the brain and something interesting. I also like it because I like when publication makes me to do some changes, right? Okay, now it's just the way of cutting the brain, but yeah, it's also kind of improvement. As I said, I'm not sure about the description of the experiments because even after really careful reading, there is still something missing, especially about this optogenetic experiment. But I mean, it was a quite good article.
Any other opinions? Ernie, did you like the paper? That's it.
Yes.
Okay. So we wrap up, right? So we don't have any questions. Thanks, everyone, for such an exciting ride. So I think I found it really interesting with all our discussions. So I wish everyone to have a really nice summertime. So if everything works out, we can continue doing this in the next months. But for now, so I wish everyone have a great time. And thanks everyone. And Olga, thank you very much. Very nice presentation and very nice explanation. Really great. Thank you, Vijay.
Thank you very much.
Yeah, thank you. Bye.
Bye bye.
Bye everyone.